Let's take a look at the fourth macromolecule. So we spoke about proteins, fats, and carbohydrates as three of the macromolecules or biomolecules that our body needs for survival. And they work in different ways. They do different things for us, like glucose or sugars or carbohydrates give us energy. Proteins, for example, they are building blocks and provide function and sometimes provide energy if we need them. And fats provide energy and also are a source of protection. But now we're talking about the fourth, which is that of nucleotides. Now we're going to talk about nucleotides, nucleic acid, and DNA. So, first thing we need to take a look at is what exactly are they? When we spoke about the other macromolecules, we said what elements they are made out of. So when we look at nucleotides, nucleic acids, and DNA, they are made up of carbons, hydrogens, oxygens, nitrogens, and phosphorus. So they're actually made up of more than the carbohydrates, which is just carbon, hydrogen, oxygen, the fats, which is just carbon, hydrogen, oxygen, the proteins, which is carbon, hydrogen, oxygen, and nitrogen. And now we've got phosphate or phosphorus forming phosphate, which is part of this whole process. You're probably sitting there thinking, who cares? What do you want about? All right, let's just take a look. Firstly, nucleotides. Nucleotides, nucleotides. What are we referring to? First thing you need to understand is that when we look at a nucleotide, it is made up of something called a nitrogenous base. Nitrogenous base. So these nitrogenous bases have this particular shape to them, like that, and they can be made out of a single ring structure, or they can a double ring structure or a single ring structure. So this is made up of nitrogen. Right, these are nitrogen rings. There's nitrogen in different aspects of this ring. And if it's two bases, an example of this is going to be adenine and guanine. Right? Adenine and guanine are two examples of double nitrogenous bases. So this could be adenine, for example, or guanine. A single base could be cytosine or thymine. Cytosine or thymine. These are the nucleotides. The nucleotides, the A's, G's, T's, and C's, right? They're these nitrogenous bases, and they're made up of nitrogen rings. They can be two rings. If they are, they're either going to be adenine or guanine, or one ring, it's going to be either cytosine or thymine. But that's not all it is. If we start looking at nucleic acids, they're not just these nitrogenous bases. They have a sugar molecule attached to them. So they're going to have a sugar molecule attached, and this sugar molecule is pretty much just like what you're going to find with glucose, for example, right? Remember, glucose, so let's just write S for sugar. Glucose is made up of carbons, hydrogens, and oxygens, right? It's a simple sugar. So is this, made up of carbons, hydrogens, and oxygens. It's just attached to the nitrogenous base. And then finally, to make up a nucleic acid, we've got a final component that's attached to it, and this is what we call the phosphate group. So there's going to be a phosphate group attached here, up here, and we know that phosphate has phosphorus in the middle with a whole bunch of oxygen on the outside, right? So you've got all this oxygen. Some of them are double bonded, some of them are single bonded. Don't stress too much about that because we just want to highlight that this is a phosphate group right here. So now what we've drawn up is our nucleic acids. So the nucleotides are simply the adenine, guanine, cytosine, thymine. They're the nitrogenous bases in green. The nucleic acids is the whole thing. Now, if you bring them together, snap them together, you start to make DNA. So again, if we do something like this, let's just draw up for the nitrogenous bases. Let's make it like this. If there's two, we're going to draw it like that. If there's one, we're going to draw it like that. So let's do that. Let's do the sugar and make it just like this. And then let's do the phosphate like this. Okay, now the phosphate group is attached to the sugar group like that, of the next one, of the one above it. So if we were to keep drawing this, for example, let's draw a single nucleotide there. Let's attach it to a sugar. Let's attach it to the phosphate. That phosphate is attached to the sugar. As you start to move down, you find that you start to create this phosphate sugar backbone. Here's the backbone. And in actual fact, you've got these nitrogenous bases here in green. They bind to other nitrogenous bases on the other side. So I said to you, if there are two nitrogenous bases, it's either adenine and guanine. Let's say that this one here is adenine, right? Let's say this one down here is cytosine. Let's say this one here is thymine. Now, if you've got adenine on one side, attached to it on the other side is going to be a thymine, a T. So let's draw up. Thymine is a single nitrogenous base. And then let's draw up the sugar attached to it. Let's draw up the phosphate attached to it. And there we go. And then what's going to be attached to a C, bound to a C? Cytosine, it's going to be guanine, a G. That's 2. So you can see when there's 2 rings, nitrogenous base, it's going to be connected to a single ring. When there's a single ring, it's going to be connected to 2 rings. So let's just write this up. That's the T, and this is the G, and then we can put the sugar here, and then the phosphate as well. And then remember I said the phosphate and the sugar form a backbone, and so they're bound to each other like that. And then finally, let's do the last one. This is going to be adenine, because wherever there's a T, there's an A, right? And then it's bound to the sugar, which is bound to the phosphate. And again, what we've created is our sugar phosphate backbone here. Now, the A and T is always connected with each other through 2 bonds, 2 hydrogen bonds. The C and G is connected through 3 hydrogen bonds, and 2 again. What we've just made here is this, right? I'm just going to put a whole new screen up. What we've made is this. An A attached to a T, a C attached to a G as an example attached to a C, a T attached to an A, and you can see that it's like this. Now, in actual fact, this ladder which we've made, which sort of looks a little bit like this, right, is twisted. And so the DNA is actually twisted around like this, and then you've got the connections like that. And there's our DNA. So in actual fact, this molecule we've drawn up is DNA. It contains information because, as you know, or as you will know, if we want to turn DNA into something functional like proteins, we need to do something called DNA transcription, where we turn DNA into RNA. Where we turn DNA into RNA, and then that RNA needs to be turned to proteins. Proteins do things in the body. So this is how DNA contains information. Now we're going to talk about this process of transcription, which is DNA to RNA, and then the process of translation, which is RNA to proteins, shortly.